Okay, I think we'll go ahead and get started as some more folks are joining. Tonight's program is Ask a Barnegat Bay Scientist, the American eel, an unappreciated hero of aquatic ecosystems. It's presented by Dr. Jim Vasslides. Jim is Barnegat Bay Partnership's Senior Program Scientist. He received his bachelor's, master's, and doctorate from Rutgers, and he's published papers on fish community assemblages, feeding interactions, early life history movements, as well as ecosystem modeling. Okay, Jim, take it away.
Great, thanks, Karen. As Karen mentioned, I'm Jim Vasslides. I'm the Senior Program Scientist for the partnership, and I've been here a little over 11 and a half years at this point, which is a pretty fantastic run, and I'm really excited to see some of our volunteers are on the webinar this evening. So hopefully, I know you guys already know all the background eel stuff that I'll talk about, but hopefully some of the data that you'll see, you'll recognize as having collected some of it. And so this should be fun for y'all. Before we get started, I just want to mention it's absolutely okay if you screenshot and share, tweet, anything that you see today. None of this is kind of proprietary. It's all information that's totally available. I just ask that if you do tweet it, if you would include my Twitter handle there on your tweets so that I can kind of see what people are saying about eels and the responses that you get. And if any questions come up, I can answer them for you. And I also want to mention, if you don't already follow the Barnegat Bay Partnership on social media, our Twitter and Facebook handles are on the right-hand side of the screen there. You really should follow us. Karen and the rest of the staff do a great job of putting fun and interesting videos of things we're doing out in the field, or just general things of interest to the ecological community. And they update it pretty regularly, and so it's a neat resource, and I think you'd really enjoy it. So be sure to check that out, and I'll have those up again at the end of this presentation as well if you miss it now. So when most people think of eels, if they think of them at all, it tends to be kind of negative, right? The bad guys from The Little Mermaid. But eels really play a critical role in aquatic systems and are important to humans as well. And this is true of most eels worldwide, but tonight I really just want to focus on one species, the American eel, which is Anguilla rostrata. What most Americans don't know is that the American eel has been part of our story, the American story, since its very beginning. Eels were an important component of Native American diets, and the Native Americans would build rock weirs along streams, and those rock weirs are still with us today. On the left image there, you can just make out kind of this rock V, this rock-shaped V, and that's a weir in the Susquehanna watershed that would capture eels as they migrated downstream, and it would funnel them into the apex of the V, where there were woven baskets that they would swim into. And the eels would then be removed from the water and would be smoked to provide food throughout the winter for the Native Americans. Eels were also a part of the first Thanksgiving, and you can see there's a kind of stylized drawing there, but there's actually a pretty well-recorded history of the Native Americans sharing eels and teaching the first colonial settlers how to fish for eels in the New World, if you will. If we fast forward 200 years into the middle of the 1800s, eels were a part of everyday life, especially in New York City. And this image is a Currier and Ives lithograph that currently hangs in the Metropolitan Museum. It's entitled Cuffy Dancing for Eels and was part of a series called Life in New York. And you can see here, this gentleman in the red is holding out a brace of eels for the gentleman in the middle here who's dancing, and the crowd is kind of gathered around and watching. So eels were, commonly consumed in New York and up and down the East Coast at the time. But despite American eels having been important to North Americans since our earliest inhabitants, we are still learning new information about their life history and the roles they play in maintaining our ecosystems. So here I have 5 statements about eels. And what I'd like for you all to do now, if you don't mind, is in the chat, indicate which of these 5 you think may not be true. And by the end of my presentation, you'll know which of these are baloney and which are the real McCoy. So I'll give everybody a second or two to read them, and you can just number them 1 through 5, the top down to the bottom. Okay. So while y'all are thinking about that a little bit, to understand why eels are so important to ecosystems, we first need to know a little bit more about their lives, because it's not your standard fish story, your fish tale, if you will. To start with, eels are diagenous, meaning that they move between 2 waters, di meaning 2, agenous, waters. They move between freshwater and saltwater. And so you are likely familiar with some other diagenous fish like river herring, sturgeon, and of course the showboat salmon there that everybody likes to see pictures of jumping up streams. But unlike those salmon, which spawn in freshwater and live their adult life stage in the ocean, which is known as anagenous, eels are patagenous, meaning they're spawned in the ocean and then live their lives in freshwater, so the opposite of what salmon do. So this is a great map from the Parks Canada, because eels are found from all the way down in the Yucatan Peninsula off of Mexico, up the Gulf Coast, up the Atlantic coast, and up into Newfoundland in northern Canada. And all of this is considered one large population. And this is because all American eels are born in the Sargasso Sea, which lies between Bermuda and Bahamas. They use oceanic currents to migrate up the Atlantic coast, so they might catch the Antilles current or the Caribbean current, the Florida current, and then hitch a ride on the Gulf Stream, up and around. And they find their way into rivers where they grow to adulthood, and then they migrate back out to the Sargasso Sea to spawn, and there they die. Now, if I told you that statement about their life history 10 to 15 years ago, it would have been accurate. And now I'm maybe half right saying that if I'm lucky. And so we'll talk a little bit more about that. The current hypothesis about the beginning of the eel life cycle is that eels spawn in the Sargasso Sea. So this is the green circle here is where they think their American eels spawn. But they also think that there's possibly a second location where American eels spawn now along this mid-Atlantic ridge, which is this white dashed rectangle during the winter, and it's during the winter and the spring. And I say that this is a hypothesis. We think they spawn in the green circle, and now we think also in this white box, because no one's actually seen eels spawning in the wild. The only evidence we have of where eels spawn are collections by boat of very early life history stages. But with the improvements in technology and science techniques and remote sensing, researchers have collected these very young eels, and typically they are part of research cruises around this area. They've collected these very young eels, determined their approximate age, and then used computer models of the ocean dynamics, and they run those models backwards. So they know what the conditions were here when they caught the eels. They think they know about how old the eels are, and so they run the computer models backwards to see where the eels would have started when they reach that age 0. And so that's where they've come up with the idea of these 2 disparate spawning areas. So I mentioned they catch these very young eels. Did you know that the newly hatched eels are invisible? Well, invisible is a bit of an exaggeration, but as you see in the picture here, the first life history stage of the eel, which is known as Leptocephalus or Leptocephali, is a long, translucent, flat ribbon with fangs. It's kind of hard to make out, but you can kind of see if your video graphics are good. They have kind of little, very sharp pin fangs sticking out there. And at this stage, they're often referred to as leaf-like because of that very flat, elongated nature. And so the Leptocephalus stage lasts for around 7 to 9 months. And during this time, the eels drift and swim with the Gulf Stream along the Atlantic coast of the US and Florida. Once the leptocephali reach about 55 to 65 millimeters in length, so step C here in the drawing, they begin their metamorphosis to their next life history stage. Their bodies begin to shrink in length and they thicken, they lose their fangs. And you see this happening in steps D, E, and F here. But they still remain transparent and are now known as glass eels. The picture on the left is someone holding kind of a handful of glass eels that they've recently caught. And you can see that they're mostly transparent with the exception of maybe this kind of thin black line that runs along their midline or their dorsal surface, depending. It's at this stage that the eels swim inland from coastal waters and begin to enter their estuaries. And depending on where you are along the coast, this can start anywhere from January in the southeast to April up in New England and Canada. And this year, our eel sampling program was catching glass eels in the first week of February. And since we were already catching them by kind of a couple at a time, it's likely they were arriving here even a little bit earlier than that, maybe the middle of January. As they enter the estuaries, eels begin to develop pigment along their bodies, starting along the top or the dorsal surface. So that was the kind of dark line that you saw in that previous slide. And the pigmentation progresses in a regular pattern down the body, and that allows us to classify eels into several sub-stages. And so here, stage 1 is what everyone would agree is a glass eel. You can see the kind of myomeres or the muscle structure of these chevrons, but they're mostly translucent. Stage 2 is when they first start getting their pigment. Like I said, it fills in at the top. So you can see just above the little red dot here, there's some stippling, some little dark dots. Stage 3, the dots extend a little bit further down and maybe even, a quarter of the way down the body. Stage 4, the dots extend down to the midline here and are filling in a bit more. Stage 5, filled in more in the upper half and extending a little past the midline. 6, even further down until they reach stage 7, which is when they're mostly pigmented along the upper 2/3 of the body. However, there's a lot of confusion regarding what to call the eels at this stage and within these substages. Some folks refer to any eel that is not fully pigmented, so anywhere from stage 1 to stage 6 as a glass eel, and a fully pigmented eel, stage 7, as an elver. Others argue that the elver stage starts when pigmentation begins. So stage 1 would be your glass eel, stage 2 through 7 would be elvers. But really, no matter what you call it, because the stages 2 through 7 don't last very long, this elver stage lasts anywhere from 3 months to a full year. So what happens after the elvers? Well, it was originally thought that the elvers continued their journey into freshwater systems, where they then entered the third phase of their life history, known as the yellow eel, due to the yellowish tinge on most of their bodies. So you can see it's kind of greenish yellow, and then cream or a little bit yellow on their bellies. But research over the past 15 years has indicated to us that not all elvers continue into fresh waters. So again, this is that second half of my earlier statement that may or may no longer be true. Some elvers remain in estuaries their entire life, maturing into yellow eels and going the entire time without entering fresh water. They stay in the estuary, they grow, they reach maturity, and then they leave. Some elvers enter freshwater for 2 to 3 years and then return to brackish waters. And then they'll make forays back and forth between each, depending on water quality and resource availability. They'll go where the water's the best and the food is the most plentiful, depending on the time of year. And some elvers move upstream for their yellow eel stage. Now, each of these strategies have trade-offs. The estuarine residents typically grow faster. There's more food, usually, in the estuaries, so they grow faster, they mature earlier, and they'll migrate downstream at a younger age. While the freshwater residents, the one that continue to swim upstream and live their entire lives in the freshwater, that leads to later maturation. So they don't reach sexual maturity until a later age, but they also reach overall larger sizes. They grow a little bit slower, but they grow throughout a longer life stage or longer life history. The yellow eel stage is also when sex differentiation occurs. And so eels at the elver stage and at the kind of early yellow stage are non-sex differentiated. And then as they grow through the yellow eel stage, they'll develop into either males or females. There are several competing hypotheses to explain sex determination, but none have really proven conclusive yet. Right now, the evidence seems to support what's called density-driven differentiation, where higher densities or more eels in a kind of smaller area lead to males and less females, more males and less females. And so those are typically in the estuary. So you get younger, quicker growing males in estuaries, where in freshwater, you tend to get older, larger females, which is actually good for kind of species propagation because the older, larger females tend to have bigger eggs and more eggs in reserve for when they actually do go to spawn. But you can have males in freshwater and females in the estuary. And so there's that balance, it's a portfolio of different sex and maturation rates and life stages and life history styles. Now, I mentioned that some eels move upstream, and I mean really far upstream they can go. Eels have been captured over 300 miles up the Delaware rivers and its headwaters. And this distribution map of New York, I think, is a great example. You can see that eels have moved up the Hudson River, which would be this kind of green block right here, and then extending up this way, generally right up to here. And green on this map is, they've sampled them after 1976. Gray are only samples from before 1976. So up the Delaware, or I'm sorry, up the Hudson. Also up the Delaware. So if you imagine the Delaware River coming down this side of New Jersey, up the Delaware here and here. And they've also gone up the Susquehanna. Historically, that would be these gray dots out this way. And if you think about the Susquehanna is the headwaters for the Chesapeake Bay. So that runs all the way down through Pennsylvania, out into Maryland, and then Delaware, and the bay down through Maryland and Delaware. And so that's quite a haul for eels to get up. And you'll also see that they've been caught off of the St. Lawrence River and even into the Great Lakes. And so the eels can really get around when they want to. There's also substantial evidence that besides this really long migration, the yellow eels establish home ranges that can be anywhere from about an acre in size in the estuaries up to 2.5 miles in length along rivers. And so they'll, folks have tagged eels and then gone back and recaptured them over time, or tracked them if they've used kind of the radio telemetry and see where the eels go. And like a lot of creatures, eels prefer to just kind of stay in one area, haunt one area that they know, and if resources are good, they'll stay there for their lives. Not different than people, I guess. So after anywhere from 5 to up to 30 years, the yellow eels become the silver eel. And silver eels are the sexually mature terminal life stage for the American eel. The age at which they reach full sexual maturity varies. It varies by latitude, by sex, and by habitat, like we talked about just a couple minutes ago, estuary versus freshwater. And they've actually had records of the oldest females being estimated at about 42 years of age. As the eels reach sexual maturity, they do undergo several physiological changes. The most obvious, which you see here, is they turn that dark gray or silver. They're no longer kind of the greenish yellow, and even their lower bodies kind of start to silver up as well. So that's the obvious ones. Some less obvious ones, their pectoral fins grow larger so that they have improved swimming speed for the long transoceanic haul. Their eyes also grow larger and change in pigment color so that they are better able to see in the ocean systems at the depths at which they're going to be traveling at. Downstream migration generally begins in the fall. It starts earlier in the northern latitudes and later in the southern latitudes. And it's thought that this latitudinal delay in migration allows the eels to all meet at the spawning grounds in the Sargasso or the Mid-Atlantic Ridge at about the same time. The other interesting thing about silver eels is that we're pretty sure that they stop eating once they begin their oceanic leg of their migration, as their digestive systems basically disintegrate. It's thought that by the time the eels reach the Sargasso Sea, they're basically emaciated, they're no longer eating, they're just kind of a tube of gonads ready to spawn, and then they die. So with that somewhat morbid thought, we now know all about the fascinating life history of American eels. And so now I'd like to talk a little bit about why the eels are so important to our ecosystems, why they're kind of the underappreciated heroes. Eels serve as a pretty popular prey item for other species. If you're a recreational saltwater fisherman, right now you've probably got one thing on your mind, and that's the striped bass migration, right? Striped bass are currently moving through the coastal waters and estuaries, and you better believe that they're cued in on the fact that there are all these small, very tasty elvers swimming around in mass trying to get up into the rivers. Studies have found that elvers are an important prey item to these migrating striped bass, and in addition, they're preyed upon by most piscivorous or fish-eating fish as they move through the estuary and then upstream through the spring and the summer. Right, they're bite-sized. They don't have any sharp spines or kind of protective spines on their opercles that cause you, if you're a fish, to get stuck on. And so they're basically kind of a jelly snack. They represent an important pulse of nutrition into the ecosystem, right? During the winter time now, there really isn't a lot of nutrients kind of coming in. And so this is a big flush of nutrients into the system in the spring, right? As things are kind of waking up, and through the summer as fish are trying to grow and get larger. And like I said, not just fish, birds. As you see, there's a cormorant in the bottom right picture. In the upper right picture, that is indeed a snake eating an eel. And they're also fed on by otters. That's a popular picture you'll see if you look up eels as prey or something. There are lots of videos of otters eating eels as well. And so the eels are a very important link in the food webs of most ecosystems. But they're not important just as a prey item. They're also very important as predators. The Leptocephalus stage, not really. That eats marine snow, just kind of debris and detritus, very small pieces that are floating around in the ocean. And the elvers, when they first get into the system, probably are eating very, very small detritus, plant material, and those sorts of things. But as they grow and get bigger, they can become very important predators in these freshwater estuarine systems. In freshwater systems especially, it's been estimated that eels can make up almost 25% of all of the fish biomass in a freshwater stream, and they eat a really wide variety of prey items. Kind of got two columns here. Column on the left are kind of the larger yellow eels. Column on the right would be your elvers and small yellow eels. The smaller eels tend to eat smaller things like fish eggs, larval fish, and lots of insects. You'll notice they're predominantly insectivores. While the larger eels eat juvenile fish, crayfish, snails, other eels. In the estuarine systems, they'll eat gastropods, they'll eat crabs, they'll also eat bivalves. Basically, if it will fit into their mouth, they'll eat it. People think of tiger sharks, right, as eating everything. It just happens that they eat bigger things. Eels eat everything. But what's I found really interesting and kind of outside of the box from a predator standpoint, there's some experimental evidence, and it's actually out of Pennsylvania, that predation by eels keeps non-native crayfish from establishing new territories or eliminates them from areas they've already invaded. These non-natives may not be used to seeing eels, and so they're not as wary of them as they should be, and so the eels preferentially snack on them over the native crayfish. And actually help to kind of keep the balance in the system. So eels serve as biological controls in the ecosystems in which they're present. Eels also are hosts, or you could think of them maybe as kind of habitat, right? Eels are key parts of the life history cycle for other aquatic organisms, not just as a prey or as a predator, but freshwater mussels, like the eastern elliptio on the left, are an important component of river ecosystems, where they improve water quality by filtering algae, sediment, and nutrients from the water column. The mussels have a kind of different life history stage also, where their larvae, which are known as glochidia, have to attach into a fish host, and they're parasitic for 2 to 4 weeks. And so eels are actually a favored host for freshwater mussel larvae. They get into the gills, which is what you see in this image on the right. These dots are all glochidia, or freshwater mussel larvae that are attached and feeding on the American eels. They'll stay there for, like I said, 2 to 4 weeks before they'll drop off of the eel gills and settle into the river substrates. Freshwater mussels are considered one of the most endangered groups of organisms in North America. And the decline in abundance in many freshwater mussels is thought to be linked to the decline in eel abundance and the contraction of eel ranges. And so they're a key component in that system. And finally, as I mentioned at the beginning of this presentation, eels are a food source for humans. While consuming eels may no longer be popular in America, eels are a prized food in some European and Asian cuisines. On the upper left here is a picture of unagi, which is a Japanese grilled or broiled eel. And on the bottom left is a jellied eel, which was very popular for some time in the United Kingdom. During the 1970s to the 1980s, if you look on this graph on the right, which would be kind of this time period here, 1970s, 1980s, there was a significant commercial fishery for the yellow and silver stage American eel, with the landings ranging from about 2.5 to about 3.6 million pounds. Now, since 1987, the landings have dropped. It's down to about 1.6 million pounds. And then it's generally been lower since then. You can see kind of the decline here, with around 781,000 pounds out here at the 216, 218, which is the second lowest landing amount since the turn of the century. Actually goes off the graph here a little. Only, I guess 2003 was about as low. However, you'll notice this brown line here, which is the value in dollars of the American eel fishery, that the values generally increased since 1970, despite this decrease since the 1990s. And I'm sure most of you immediately noticed this dramatic jump over here in 2011, when the fishery value went from about $3.5 million to over $40 million. And you're probably wondering what's going on. Well, in 2010, the European Union banned the export of live glass eels out of their member nations. And this had a pretty substantial ripple effect across eel fisheries worldwide. So let me explain. No, there is too much. Let me sum up. Because no one has been able to breed eels in captivity, eel aquaculture consists of the capture of wild glass eels and elvers, and then taking them from the wild, bringing them back to an aquaculture facility and growing them into adult eels before they're sold into the commercial food market. As the populations of first the Japanese and then the European eels crashed, which led to the export moratorium, the demand for American eels to export to these aquaculture markets has increased. The demand for eels, elvers and juveniles, or elvers and glass eels, is actually so high that in 2019, a pound of glass eels was worth $3,000, which was more than 10 times the price of the same weight of silver. And believe it or not, in the United States, only 2 states, Maine and South Carolina, have approved elver or glass eel fisheries. So there are only 2 locations that people could get them from, basically to export out of the United States. Europe was closed, and Japan had very few eels that they were able to collect in the wild. So, as you can imagine, as the demand for and the price of elvers skyrocketed, there was a pretty immediate response in the black market. Yes, people were black market buying and selling coolers full of glass eels to ship overseas. Between 2011 and 2014, the U.S. Fish and Wildlife Service ran an undercover eel poaching operation called Operation Broken Glass. I kid you not, that was the name of it. By the time things were all said and done in 2018, 21 individuals were doing jail time and or had probation, and there were over $350,000 in fines, forfeitures, and restitution. And that was just over the course of 3 years. And that at that time, glass eels were only selling for, $1,500, $1,800 a pound, only $1,500 to $1,800 a pound. Now it's $3,000 a pound. So I suspect, right, it hasn't really, I'm sure there's still some of that going on. And New Jersey certainly has not been immune to this problem. As part of the sting operation in 2013, the New Jersey Department of Environmental Protection conservation officers caught 3 men on an Atlantic County Utilities Authority property down in Absecon, adjacent to Absecon Creek, fishing with an illegal flake net. They found them in the middle of the night. It actually wasn't too far off the road, and apparently wasn't too terribly hard to see them breaking the law. But they kind of knew they were going to be there anyway. The men were arrested, their gear was confiscated, and because they had holding tanks set up in the back of their pickup trucks, their vehicles were also subject to forfeiture. So they basically got everything that those guys had. So we've talked a bit about how important eels are to the environment, and I've also alluded to a decrease in their abundance and range compared to their historic population. but I thought we would just touch kind of quickly on how eel populations are doing now. The figure on the left is an index of abundance, so kind of a idea of how the yellow stage eels have been doing on the US Atlantic coast from 1974 to 2016. And this information is collected by the individual states and all brought to kind of a coastwide management agency. While the figure on the right is an index of abundance for glass eels and elvers over a shorter time frame from 1988 to 2013 actually. And kind of I'll give you the takeaways here. For the yellow eels, the population declined through the first part of the period, roughly the first 20 years, so 74 to about 94, you can see a general decline, and then has remained relatively stable since then, bouncing up and down a little bit, but not any really big changes. For glass eels and elvers, there really hasn't been any discernible trend since the late 1980s, but you'll notice right away that there's lots of variability, right? It goes down and spikes back up, down, up, down, up, down. And this annual variability is normal for indices of early life histories of fishes. As you can imagine, when you're small, tasty, and at the whim of the environment, you suffer pretty high mortality. So boom years, and bust periods can be common. However, because of the low abundances of eels compared to their presumed historic levels, over the past 20 years, they've actually twice been considered for listing under the Endangered Species Act, once in 2004 and once in 2010. Now, both assessments concluded that while low, the population is stable and not in danger of immediate extinction. But they did recommend that measures be taken to reduce mortality and improve conditions along the coast for eels. And in their assessment reports, the federal agencies recognized a number of obstacles to rebuilding eel populations. Amongst them, one of the main concerns is habitat loss or blockages of river passage. While American eels are thought to be habitat generalists, right? They like the estuaries, they can live in fresh water, they can tolerate a wide range of water quality conditions. They still need to be able to access that habitat. And, since the 1800s, let's face it, our rivers have been fragmented with the proliferation of dams. The map on the left is a distribution of larger dams, so generally 6 feet or higher, each of those red dots. Within, I clipped it to kind of the eastern half of the United States. A somewhat recent study, had counted over 15,000 dams within the range of eels. And each of these dams pose an impediment to eels trying to move upstream and downstream. And so that same assessment that had estimated that kind of 15,000 dams within eel habitat also suggested that stream habitat for American eels has been reduced over 80% from about 346,000 stream miles available to eels down to about 56,000 miles available to eels. And dam blockages, or blockages due to dams, are recognized as such a problem that eel ramps, like the one on the left here, are often mandated as part of hydropower dam relicensing permits. And the idea is that if you provide kind of a more gentle incline and some appropriate wet substrate, that eels will prefer to wriggle their way up this and over top of the dam and drop in on the upstream side versus trying to go up the face of a dam. With all of the concerns about decline in American eels and fragmentation of their habitat coastwide, the Barnegat Bay Partnership and our partners were interested in understanding the status and needs of eels within the Barnegat Bay watershed. And so our first foray into kind of eel research began over a decade ago. where our partners decided to fund the Rutgers University Marine Field Station to investigate the feasibility of using kind of low-cost passage structures to get elvers over dams in the watershed. The previous picture was kind of a highly engineered structure to get eels up over a fairly large hydroelectric dam. We don't really have those in our watershed, but we have lots of those kind of small to medium dams, right? 4 to 8 to 10 feet tall. So is there a low cost way to get the dam or to get the eels that are coming up to those dams up over those? And so the investigators from the Rutgers Field Station worked at dams on the Tuckerton Creek, Tuckerton Creek Dam, the Mill Creek Dam, and then up here at the Kettle Creek Dam. And they use combinations of burlap, like you would a burlap sack, used mesh from some of their fishing trawls, some of their research fishing trawls, and then corrugated plastic pipes to assess eel preference for passage materials. And so the eels generally preferred moving through trawl mesh inside of piping compared to burlap laid across the dam face. And so in this photograph here, hopefully you can make it out, there's kind of this white PVC pipe, and then there's a black corrugated pipe that runs along here and then kind of up the dam, and they were able to suspend the pipe to make a nice slope. And this pipe is packed full of kind of net that has a mesh, a large mesh in it, and water from the lake up above the dam flows through it, and the eels wiggle their way up through that. And so they were fairly successful with that. And so we know that is one mechanism that we can use to get eels. If we identify a dam that has a lot of eels at the bottom of it that don't seem to be passing, we can get them up over the top. The BBP has also been monitoring juvenile eel ingress at several waterways in our watershed since 2012. Using volunteer constructed eel collectors. And if any of you helped build these eel collectors, the photos in the upper right, thank you very much. We appreciate it. We've been counting and staging glass eels and elvers at the dam at Lake Shenandoah, so up here, a little bit more inland. The dam at Kettle Creek, we continued studying that waterway. Long Swamp Creek, which is off of the Toms River. And then the Parker Avenue Dam down here in Forked River. And during the course of 9 years at those 4 locations, we've set over 2,000 collectors and we've captured over 31,000 eels. And for those of you wondering about how you stage an eel into those 7 categories that I showed you way back towards the beginning with those little stippling dots, well, you knock them out with a tranquilizer and then you put them under a microscope where they look like this image in the lower right. And here's his face, his heart. Gill structure. I say his, but it could be undifferentiated sex eel. And then you can see that maybe you can make out that there's just a little bit of stippling on the top of the eel up above their vertebrae there. Ideally, we would read them a little bit further back here, but the middle of the body doesn't make for a very nice picture where this nice smiling face certainly does. So now just one slide of data for those of you who joined to kind of hear about the data. There's only one slide of it. And I haven't really had a chance to digest all of the data we have from the last 9 years, but there certainly is some basic information that we can glean from it. This bar plot on the left displays the yearly mean catch per unit effort at each site. And so for each site, for each year, we added up all the eels that we caught at that site in that year and divided it by the number of collectors that we put out at that site in that year. I want to point out right away that the, oh, my laser went away. There we go. That the axes here are different scales. So pay attention a little bit to the numbers. But what you'll notice is that the Kettle Creek generally had the highest catch per unit efforts. You see their scale goes, in jumps of 30 all the way up to 90, followed by Long Swamp Creek down here at the bottom, 0 to 20, Forked River up at the top, 0 to 6, and finally Lake Shenandoah, which was 0 to 2. But there's really no clear pattern in the yearly catch per unit efforts across all sites. So in other words, the highest catches per unit efforts at one site didn't occur in the same year as the highest catch per unit effort in another site. Here, Long Swamp Creek was highest in 2016, where that was, amongst the lowest at Lake Shenandoah in that year, and, kind of amongst the lowest at Forked River in that year. And so no real clear pattern there. You do notice, though, that the yearly catch per unit efforts were generally in the same range at each site. So no real kind of lows to then a crazy high spike, except for Kettle Creek here, which saw a really big jump in 2019. And so if you kind of shift over to the figure on the right, this shows the weekly mean catch per unit effort at each site. So now our x-axis are the weeks, starting with the 5th week of the year, so kind of the beginning of February, to the 21st week of the year, which is the May, middle of May. And now the same sites, Forked River, Kettle Creek, Shenandoah, and Long Swamp, from top to bottom. And now the lines are the years, so same color across both maps there. And so if you look at this kind of finer scale breakdown over time, you'll notice that Kettle Creek, this big abundance boom in 2019, is really associated with just one spike. It really jumped in 1 week, right at week 15. Huge surge we saw. But you also notice that in general, we get our biggest abundances between weeks 14 and 16, which is kind of the first couple of, first 2 weeks in April. And so that's when we typically see our biggest arrivals. So kind of the timing generally consistent across years, though not entirely. Again, Long Swamp Creek, throwing that off with that 2016 number, and 2018, a little bit earlier than some of the other sites. So more insights to be gathered from this data as I'm able to spend more time with it. So keep an eye out on our social media, as I mentioned, because as we delve into this over the coming months, hopefully there'll be some really interesting tidbits that come out of it. And we plan on continuing this project for as long as we're able. Our intent is that this data will be included as part of the Coastwide Juvenile Eel Index that you saw just a couple of slides ago, that get used to help decide how many eels should be harvested in any given year, and what the kind of overall status of the eel population is. So besides just showing you this information during these kind of webinars and publishing it in scientific journals, it actually will go toward management purposes, which is really exciting because there aren't a lot of juvenile eel indexes out there. So now we circle back to the 5 statements I showed at the beginning of my talk. And so I've shown that the first 4 are true. And it looks like a couple of you responded back. And so it was varied about which ones people thought were true. But I've shown that the first 4 are true. So that means the last one must be false, right? Well, not exactly. Where'd my pointer go? Oh, things were going so well. Here we go. So eels can climb vertical walls as long as they're a little rough and a little moist. Elvers will find their way up. And so this is just some video during the day, and you can see them kind of, wriggling and grabbing onto all the little nooks and crannies they can. I've had state eel biologists who've told me they've gone out in the evenings to barriers like this, where they have good eel populations, and at night it looks like the entire wall, when they shine their headlamps on it, looks like the entire wall is moving because it's just thousands of elvers climbing up, trying to get up into the water. So pretty cool. If you ever happen to be out near a waterway that has elvers and a dam in the evening, take a look. So that is all I have for you this evening. Thank you so much for tuning in, And Karen, if we have time, I will take some questions. If you have a question and I don't get to it tonight, please feel free to email me. My email is my first initial and last name at ocean.edu. If you prefer to tweet at me, tweet at me. And again, be sure to follow the Barnegat Bay Partnership social media sites because we do fun things.
So there is one question, Jim.
Oh, great.
John is asking how many ladders are actually located in ratio to dams in New Jersey. So he's talking about the whole state.
Oh, that's a great question. I'm going to say it's very, very, very low. I mean, I'm mostly familiar with the Barnegat Bay watershed, and we don't have any eel-specific ladders. There are some fish ladders, and eels will use fish ladders, but they're not really designed for eels. The flow might be a little too high. We don't have any eel-specific eelways, they call them, or eel ladders in our watershed. There may be one or two in other places. I have heard of some folks throwing the burlap sacks over the face of the dams to try and get eels into those, to climb up over those. But I don't think that was really a kind of, purposeful, permitted sort of activity. So.
Okay, the questions are rolling in. So Bill is asking, is there any relationship between the freshwater where a parent eel lived and where their progeny end up, or is it a more random function of currents and chance?
Great question, Bill. So eels are considered a panmictic population, it's called. They all spawn together in the same area. And then those Leptocephalus, they just kind of drift, and swim a little bit up the Gulf Stream, and they end up by chance. They smell fresh water, basically. They have, a good olfactory sense. And, when they are far enough along in their metamorphosis that their swimming ability gets up there, they'll turn their head in and swim in. So yeah, totally by chance. So you could have a male donor from Newfoundland, Canada, and a female donor from South Carolina, and the juvenile eel could end up in Maryland.
How long have you been studying eels? Someone wants to know.
Yeah, well, so I have been interested in eels before I got to the Barnegat Bay Partnership. So 13, 14 years, but I was really able to kind of dive into it once I got here. Kind of spurred on by some of the work that the folks at Rutgers did with that ladder. And so I had a colleague who was working with eels, Mark Sullivan, who's now at Stockton University, Dr. Mark Sullivan, who had kind of first shown me those eel collectors, and they were low cost, and BBP likes things that are low cost. And so, we had some volunteers help us out and make up a bunch, and we've been doing it for, over 9 years now.
How do mature eels get back over the dams when they're going to back to the bay, to saltwater?
Sure. Parachutes. No, I'm kidding. Yeah, they just launch themselves down over the front, and so I focus mostly on upstream migration, but downstream migration is just a bigger deal, going over dams, and actually for hydroelectric dams potentially more so, because if you think about it, when you're small, if you can, you have a better chance of I shouldn't say better. You are less likely to be hit by a spinning turbine because you're little, but when you're a big eel coming through a hydroelectric dam, you're more likely to get chopped up into little pieces. So that is a big concern, both for hydroelectric dams and for smaller dams as well, but less so for the smaller ones. Eels are incredibly resilient when they get large like that. And they can actually kind of move over land a little bit as well for some period of time, as long as they don't dry out. And so they'll typically, try and find their way back down over the face of the dam with the flow of water.
What is the decline in Barnegat Bay over the last decades and why, in reference to eels?
Yeah, well, and so, if I haven't had a chance to really dive into the numbers yet, but just kind of eyeballing them and just to keep those graphs from being too cluttered, I didn't actually show kind of the variance or variation, the standard deviation with that data. But if you kind of looked at it instead of just straight bars as more of a pattern, you'd see that even within each year, it's very kind of drawn out. And so there may not actually, I suspect in Barnegat Bay, it's relatively steady. We wouldn't be able to say that from our catches, we've seen a decline bay-wide. Some of the sites we may have, and if that's the case, if we're seeing it, not necessarily bay-wide, but at one location versus others, that's probably a better indicator of a more localized habitat issue. Maybe there was some blockage or something happened downstream of where we're collecting that's keeping them from getting up to our point. That could be. I've seen some other research in Barnegat Bay where they were getting lots and lots of eels over the course of a couple of years at like the Tuckerton Dam, but then the Mill Creek around Stafford Township at the dam there by Route 9, they weren't getting any eels, and this was out of Rutgers, and they kind of supposed that there are a lot of those dead-end lagoon communities between the mouth of Mill Creek and the dam, and the eels are just going up those and getting lost. And so that's more of a localized habitat thing, not necessarily a true decline, but just a decline at that one location.
Do you have a recommendation for eel educational video resources, similar to the PBS documentary Mystery of the Eels, and then geared toward high school students? They added. I guess this is a teacher.
Yeah, I don't, but I bet you Karen and I could come up with something. I know, there are certainly no shortage of eel videos, and I suspect actually maybe, sorry, Karen, I'm like, you're up here on this screen for me. Chris Bowser at New York State DEC, who is also at what's the Hudson National Estuarine Research Reserve? Hudson River National Estuarine Research Reserve actually does a lot of education and outreach, community education outreach using eels. And so he might be a really good source. If you want to send me an email to my email address there, I can kind of send you Chris's way. And I know I've seen videos he's done that he's shown at scientific meetings that people have been really impressed by. So that might be a good one.
Okay, John is asking, what's the most effective way to catch eels if someone was to use them for fishing?
So using for fishing would probably be like a yellow eel. And that's typically kind of the small yellow eels, right, are used for striped bass bait or other kind of large game fish. The most effective way would be probably whatever you can do under New Jersey state law, which I think for recreational fishermen, and don't quote me on this one because I haven't looked at the regs in a while, maybe potting, but I'm not positive. But in the Fish and Wildlife Digest, there are specific regulations for what gear you may use and how many eels you can keep, because there is a limit for both recreational and commercial fishermen.
Okay, question from Bill. Is there any difference in the food in the Sargasso Sea versus the Atlantic Ridge that could be detected in the eels and support the idea that there are two different breeding areas?
There probably is, but they don't really eat at the Leptocephalus stage. It's marine snow, and I'll have to admit, things that you need to look at under really high-powered microscopes are not my favorite. And so I don't really know that much about kind of the marine debris, marine snow at that size. So I don't know. That's a great question, Bill. But yeah, I mean, leptocephali, that stuff breaks down. Generally speaking, food items that small break down super quickly. And so it's really hard to distinguish what it is. But, I would imagine folks have done, besides food, what's actually pretty popular is microchemistry on hard parts. So on the ear bones of the eels, depending on where they've been swimming, will collect different chemical markers. And so you can actually, well, not you or me, but folks can actually kind of ablate those ear bones and then measure them in a gas chromatograph and see kind of different signals. But then the trick is tying that back to a particular water mass. They do it for fish, for kind of juvenile fish. I suspect they're probably trying to do it for eels, but it's much more difficult to do than I just made it sound.
How will the impacts of climate change affect eel migration patterns?
Fantastic question. And one of the things that I've been interested in and have kind of alluded to a little bit when I said, looking at the Barnegat Bay specific data, when, I said most of the peaks in eel abundance were between, weeks 14 and 16. What you'd expect, eels really base their kind of migration, especially their peak migration runs, on water temperature. And so you'd expect if water temperatures warmed up earlier and to a sufficient degree that we would start seeing that peak in eel abundance move earlier in the year. So that was one of the things that I'm kind of looking for. And it may be that, what I showed you was kind of weekly sampling. We actually sampled twice a week. And so maybe I need to start looking at it at a finer scale. I don't know. That's kind of where I'm headed, though. That was a great question. Thank you.
Did the warmer water effluent from Oyster Creek power plant affect the eels in any way?
Great question. I don't know. The answer to that is I don't know. Based on what I just said, you'd expect that kind of eels in that area as they're coming in January may have tried to make a run up. I guess that would be Oyster Creek was the outlet one. May have, tried to make a run up Oyster Creek because the water was warmer, but if it was too warm, it may have turned them off if there's too much of a thermal difference. But no one ever had any collectors there, so I actually don't know the answer to that. But that's a good question, especially since you'd expect that probably most of the eels that we get up in our northern portion of the bay have come in either through the Little Egg or the Barnegat Inlet, and so they would have kind of made their way northward and potentially have come across that plume of warmer water that was coming out of Oyster Creek.
How has the EU restriction of exporting worked in increasing the population?
I don't know much about European eel population. To be honest, I don't know. Sorry.
How many babies are spawned from eel moms?
I wish I could honestly switch over to Google real fast and type that up. I don't know. I can tell you it will be size dependent. Bigger moms typically have more babies and larger eggs that are healthier. So it's really not a question of how many babies per mom per se, right? Because they're externally fertilized. The females put the eggs up into the water, the males swim around and just shoot milt into the water and it all mixes together. But that's one of the things that scientists look at is how many eggs are the females carrying that could be fertilized. And larger, older females up to some point typically have more larger, healthier eggs. but I don't know I stumbled across it, but unfortunately I don't have that number at my fingertips. But it's going to be a lot of fish because of that spawning method where they just shoot everything into the water. We're talking into the, hundreds of thousands easily per spawner.
How long can an eel stay out of water before its slime is ineffective or it needs to go back in the water?
Well, some of that will depend on where it's at, how moist it is. Is it getting kind of, misted or dripped on? A while, and by a while, probably, hours and hours. I've seen firsthand an eel that slipped out of a tank unbeknownst to people through grates in the floor, but was getting splashed by kind of oversplash out of the tank and was still alive a week later. So quite some time, assuming they can stay a little moist. But, if you put them out in the hot sun on the pavement, probably not very long.
Is there any anticipated dip in the eel young populations dying due to microplastic consumption as part of the detritus snow? And are you detecting plastics in the eels you are sampling?
So we don't look for plastics. And Bill, you are killing me today. Happen to know Bill. So we actually don't do stomach content analysis. We actually put all of our eels back alive. Like I said, we just tranquilize them and then wake them back up and put them back in the waterway that we captured them in. Dip in populations due to microplastic consumption as part of the marine snow. There's probably an effect. I don't know how measurable it will be given all of the other things that can happen to really small eels. Being eaten, bad water quality. So I'm not sure if we'd be able to pick that up. But it's an interesting thing, and that's the sort of thing that I'll put out on Twitter and see if I can get an answer for.
I think that might be, I'm going to check the Q&A. I think that is the last question. Although there was a comment that Ms. Nig, I'm probably pronouncing her, wants you to come to Mast. It will be fun.
Miss Ung, thank you.
Okay, sorry. Sorry for the mispronunciation.
Yeah, well, thanks everybody for coming out on a Wednesday evening to listen to me talk about eels, because I think they're super cool, and hopefully now you do too, and we'll be part of the eel army getting out there to spread the word about why they're important.
Thank you, Jim. That was awesome. And I just want to remind everybody that tomorrow we'll be putting the link to the recording up on our website, so that if you missed any part of it, or if you have a friend you think might want to see it, you can pass it along. Thanks, everybody. Have a good evening.
Thanks, everyone.